genes. The reason that my teeth look like this and the top of my head looks like this. How's it going, folks? So, today we're going to be doing a quick review going over genetics, namely Mendelian and non-Mendelian genetics. I've got a forensic case practice sheet for you that gets a little bit complicated, so I want to make sure that we have time. But first, let's talk about the AP exam. So if none of you are aware, you are going to need to go to the AP classroom and log in. They are going to be requiring you to download and use something called the Lockdown browser, which you can see up over here. Alright, that's going to be the portal that you're going to use when they assign the AP exams. There are some practice materials that I cannot assign and I can't give you until you guys have this browser downloaded and installed. It's the security measure that they're going with. That said, if you check the AP classroom and what we have, I've assigned a couple of different things to you. So in here, you are going to find, first of all, I can't find it. You're going to find that I've assigned a genetics kind of practice thing. Some of the questions are pretty rough. What they've done is they've started to put up more and more free response questions in preparation for the test. And so, some of the ones that we're seeing are fairly large, more encompassing questions, because this is what they're going to be testing you on. But I've assigned a number of those along with some chi-square multiple choice questions just so you can practice chi-square. Yes, we are finding chi-square analysis not just in Hardy-Weinberg, but also when we're talking about genetics, and especially when we're looking at the expected phenotypes compared to the phenotypes that we actually get. Very similar to Hardy-Weinberg, works the exact same way for chi-squared analysis. So I want you guys to get some practice with that. But the review is not graded. It is not timed. It is set up for you to go at your leisure, when you have time, when you are ready to push and try in order to attempt a few questions, and then maybe you come back to it later. It's not a stressful thing to do, although you may find it stressful if you don't remember a lot of the stuff, because it really does ask stuff from across the board. The only thing I'm going to suggest is that as you start working on these, don't overthink the answer. Don't sit there trying to think about what is the catch, how deep does this rabbit hole go. If it's asking you, what is the evidence that chlorophyll is responsible for some level of inheritance, what does chlorophyll do for a plant? It does photosynthesis and it makes it green. Use that information. Your answer is going to be very easy. Then think about, all right, what terms do I need to cover? What things are related to this easy answer for me to make it an AP level answer. Now, for today, before we move on, I want to point this out. So at this point, in the table of contents or in your folder in Schoology, you're going to find the Crown Jewels PDF case, whatever you want to call it. The tricky part with this is that the numbering on the pages is not the same as the numbering in the PDF. And I know that that sounds weird, but basically what it means when we open this up, if it opens up, is it doing that? Come on. Here. Alright, so when we take a look at this, in the PDF, this is page 1. But of the actual file, this one's not numbered. This one's not numbered, which means that our first page in the actual file is labeled down here. This says it is page 1. Let me bring this up so that everybody can see it better. Alright, it says it's page 1. Even though, if you look down to the PDF guide at the bottom, this is page 3. So let me show you the pages that I want you to do. Under normal circumstances, I never would have printed this whole thing. I only would have printed the parts that are important for us. So when you take a look at it, This is all background information that you're welcome to check out or not as you deem fit. A review of exactly what restriction enzymes are, that's good for you to know because that's what this lab starts with. Review of how gel electrophoresis works, good to know and remember. Our pre-lab, we're still not there yet. We're going through all of this. This will be your first page that you're going to be working on. City Police Department Police Report. This is, according to the PDF, page 12. But according to the actual files, it's page 10. So just go until you find this. Alright, here you're going to get your information about what happened. Here are your suspects, their names, their descriptions, and then we move on to the next page of this. So here you're going to create a gel electrophoresis. You're going to need to take each suspect's DNA, apply restriction enzymes to it, theoretically, and then put the fragments up. They're numbered over here on the right-hand side, and all the suspect's DNA taglines are right here. So it should be pretty straightforward. Alright, that'll be the first part. You're going to end up with this final report here. For team members, it's just going to be one person, because this is what you're going to do. Then, name the person you found the DNA from, and explain how you came to this, however you wish. If you want to reply or refer to a diagram, make sure that you tell it what figure it should be looking at. Alright, then we're going to skip. We don't need this part. We can't do this part. This part isn't terribly useful to us. This is where we're going to come back up. Page 21 on the PDF form. I think it's 18 on the actual PDF itself. And here we are taking a look at, once again, how these restriction enzymes work and the effect that they have on DNA. So it gives you the example of how it cuts, some questions for you to answer. Keep going right on to restriction enzyme worksheet 2. Same idea. Follow along with what it says. This one's going to be a little bit more specific. It's going to want you to label out each of these fragments in well B. And that will be the end of it right here. Page 22, page 24 on the bottom guide. You can ignore the rest of it. So you're only going to be doing page 12 through, I think it's 16, and then 21 through 24. Those are the only ones you need to worry about. Now, for this, because of the way that it works, my suggestion for you is going to be, feel free to do this on a separate sheet of paper, snap a picture, and send that to me. I hope Pigeonetics was fun for everyone. I really liked it. I haven't gotten anybody's email or message about how far they got. So maybe people are still pushing on it. Maybe you guys decided, I'm gonna do this, and you're just trying to, give yourself some time. Because let me tell you, once you get past about 19, some of those puzzles can get really challenging. They can take some real time. When you're looking at something that takes 3 or 4 generations, it's easy to make a single mistake at one level and need to start all over again. So hopefully I'll see that soon. I've gotten one of the cladogram evolution projects, so that's good. The other ones should be to me before the end of the day. Looking at watch. Ooh. So, for our basics though, for our basic reviews, remember the simplest forms of genetic inheritance as you start to move on and work on some of these questions. Remember the Punnett Squares, the classic heterozygous cross. When we worked with the fruit flies, this was the bane of your existences, some of you. But keep in mind that we're looking for those percentages, and that when we do any form of Punnett square, and we do some form of cross. Let me try to get this so the light isn't reflecting off of it. Alright, we should, in the case of a heterozygous, see this kind of an output where we have a 3-to-1 ratio. This one, and this one will all be dominant. And then we only have one showing the recessive phenotype, whatever that is. And you can do additional Punnett squares to kind of review this to see how else it might work. So let's say that you look at your problem, in this case for fruit flies, and it doesn't come out to a 3-to-1 ratio. Okay, well, let's find out what else it might be. What if this one was homozygous dominant and that parent was heterozygous? If you take a look, this would have all 4 dominant. We can try it over and over again. You can try every possible permutation until you find one that matches roughly the numerical observed values that you are getting from it. Additionally, you want to make sure that those numbers are valid, which means chi-square. When I see you on Monday, and good news on that regard, my Twitch suspension is officially over. They already reviewed it. They apparently agreed, not that anybody actually bothered to email me about it, and they said, yep, we're good. We can put you back online, so I can log back in. Monday we'll go back to our 12 noon, so if you guys have questions about things, you can ask me about them, and I can actually reply to you. In addition to that, on Monday we're going to go back to the fly lab for Drosophila, and we're going to have a return to it. Because if that's part of the free response questions that they're going to be asking for this year's truncated exam, I would not at all be surprised for them to drop a Drosophila question on you. So I want us to get some more time practicing with it. We'll go back through the virtual lab. I will have a particular set that I've already played through a couple of times and determined so that we know what the values should end up like, and we can use that to analyze them instead of having you guys do your own choices, and that should be helpful for hopefully illustrating and practicing some of those. Alright, with that said, people have projects to finish. Remember, you got to explain it to somebody. Make somebody else understand what that diagram is and what it's including. The pigeonetics update, how far did you get? And now the crown jewels study that we're working on today. I'm trying to orient my finger so you can only see my mouth through it. Oh, that looks kind of weird. I like it. All right, ladies and gentlemen, you have a great weekend. Stay safe, stay healthy. I'll see you live on Twitch on Monday.